As part of your AS Nav A2, you have to be able to prepare rather simple plant cell slides and animal cell slides. Firstly, I'm going to show you how to prepare a plant cell slide. Because the cells are going to be seen with a light microscope, we have to have a single layer of cells. An onion quite simply peels into different layers. Peel off a section of the onion, get our scalpel and slice a small section of the onion. And what I'm ideally trying to get is a small square of cells to go on the centre of the slide. However, if I put this onto the slide and try to view it with the light microscope, it would be too thick. Thankfully with onions, the inside layer of cells just on the lighter surface of the onion will peel off. But once again, if I put this under the light microscope, these cells are almost colourless. You wouldn't see anything. So to be able to see the cells, I'm going to have to put some stain onto them. One of the most common stains for plant cells is iodine. Now it does take a few minutes for the iodine to actually stain, so I would leave this for approximately 3 or 4 minutes before taking a thin glass cover slip and then very gently place the cover slip over the top of the cells. But what you mustn't do is press on top of the cover slip, because if you do that, you'll actually break your cells, and when you look at it under the microscope, you won't see what you're trying to find. This is how to prepare an animal cell slide. It's even easier than preparing a plant cell slide. The most easily accessible animal cells from our bodies, you might be surprised to hear, actually come from inside our mouths. So we're going to be looking at cheek cells, which are obviously from a human, which makes them an animal cell. Inside our mouths, around the gum line, the cells come off quite easily. Now, by placing a cotton bud into your mouth and rubbing it quite simply along the inside of your gum line and up along your cheek, you may not be aware of it, but actually what you're doing is you're scraping off cheek cells, and you'll also take some saliva as well. It's quite handy to get the end of your cotton bud quite damp really with saliva. It's quite nice. Take your slide and your saliva-covered cotton bud and cheek cells and rub them onto the inside or centre of your slide like that. Now all it will look like on your slide is a damp patch with maybe a few blobs on it. The few blobs that you can see are going to be the cheek cells. They're obviously see-through and you won't see them on the light microscope. So what you need to do is to take your methylene blue dye and you place a drop or two on the centre of your slide. And once again, you gently drop the cover slip over the top of the stain. And there you have your prepared animal cell slide.